Hello grade 8 students. In this video we'll continue about digestion chapter and here part 2 about the food tests. Let's start first with the first food test. We will talk about the Fehling test. What is the aim of Fehling test? Fehling test, it's used to test or to detect the presence of sugar, mainly reducing sugar in food. You have to know that all the types of sugar, they are considered as reducing except sucrose. So all of them are called reducing sugars except for sucrose, which is a non-reducing sugar. So now let's move to the procedure of the test. First of all, we have to put a piece of food inside a test tube. This piece of food is going to be tested. So we put the piece of food inside this test tube. We had to add equal amounts of Fehling A and Fehling B solutions. So Fehling A solution and Fehling B solution, we have to add equal drops. For example, if I add 2 drops of Fehling A solution, I have to add 2 drops of Fehling B solution. If 3 drops, 3 drops, and so on. The obtained solution, Fehling solution, has blue color. So this solution has a blue color. We have to heat gently the test tube over the Bunsen burner. Bunsen burner is the source of heat. So Bunsen burner is the source of heat. This is found here, a source of heat. So we repeat this. First of all, we have to put a piece of food to be tested inside the test tube, as shown here. Add equal drops or equal amounts of Fehling A and Fehling B solutions to the test tube. You have to know that Fehling solution has a blue color. We will heat the test tube gently over a Bunsen burner, which is the source of heat. So step 1, put the food in the test tube to be tested. Number 2, add equal drops. And number 3, heat it gently over a Bunsen burner. Obtained results. We have a positive result, we have a negative result. From now on, keep it in your mind. Whenever we have positive, meaning that we have the substance. It's negative, we don't have the substance. Positive result with Fehling test, appearance of a precipitate. It's brick red precipitate as shown in this figure. So appearance of the brick red precipitate, this color is called brick red. Okay, so appearance of the brick red precipitate indicates the presence of reducing sugar. So this is called a positive result. What is the negative result? The negative result is the no change in color will appear. So the color will remain blue as the color of the Fehling solution. So whenever we have a blue color, so this is a negative result. Okay, so there is no reducing sugar. So I repeat, first of all, we said that we have procedure for the Fehling solution test. First, we put the food in a test tube to be tested. We add equal drops or equal amounts of Fehling A and Fehling B. Note that the Fehling solution has a blue color. Number 3, we have to heat the test tube gently over a Bunsen burner. If it's a positive result, we will have brick red precipitate. This brick red precipitate, these we have reducing sugar. If we had the brick red precipitate, which is the positive result. While if the color remains blue, no change in color, this means that absence of reducing sugar. So there is no reducing sugar. So as a summary for the first test, test number 1, Fehling test. Fehling test is done to identify the presence of reducing sugar. We said that all the types of sugar, they are reducing except for sucrose, which is a non-reducing sugar. We have to know that the procedure, we have to put a piece of food in a test tube. Then we had to add equal amount of phenyl A and phenyl B. The solution has a blue color. It's called phthalate solution. Then step number 3, we have to heat the test tube gently over the Bunsen burner, which is the source of heat. If it's a positive result, we have brick red precipitate. If it's a negative result, the color remains blue. So positive result, brick red precipitate, meaning that we have reducing sugar. If there is no reducing sugar, it's a negative result and the color remains blue. Now let's move to another test. So first test, it was Fehling test. It's done to identify the presence of reducing sugar. Moving to iodine test. Iodine test, the aim of this test, it's done to test the presence of starch. So iodine, it's done to identify the presence of starch. What is the procedure? Number 1, we have to put a piece of food to be tested in a small dish. Then add few drops of iodine solution. This is step number 2. The solution has brown to orange color, the iodine solution. What are the results? If we have a positive result, we have dark blue color. So this dark blue color indicates the presence of starch. So here we have starch. While the negative result, no change in the color. So we have brown orange color. So this indicates the absence of starch. Here there is no starch since they have brown orange color. So if we want to analyze this, how can we analyze? So after adding iodine solution to the food, the color gives us a dark blue color. If we have to interpret, we have to add this indicates that the food contains starch. Or for the negative, after adding iodine solution to the food, there is no dark blue color and the color remains brown to orange. So what does this indicate? This indicates that the food does not contain starch, or this indicates the absence of starch. So till now we have 2 tests. The first test is called the Fehling test. It's done to identify the presence of reducing sugar. The second test is called the iodine test, and it's done to identify the presence of starch. Let's move now to test number 3. Test number 3 is called Biuret test. Biuret test is done to identify the presence of proteins and or polypeptides. So we have proteins and polypeptides, or we have proteins or polypeptides. So here is for proteins and / or. Either we have protein and polypeptide together, or we have proteins or we have polypeptides. So procedure, what do we have to do? We have to put a piece of food in a test tube. This is the first step. Second, we have to add equal drops of copper sulfate. What is copper sulfate? It's the CuSO4 and sodium hydroxide. So copper sulfate, CuSO4. Sodium hydroxide is the NaOH. So this is number 2. Number 1, put a piece of food in a test tube, add equal volumes of copper sulfate, CuSO4, and sodium hydroxide, NaOH. So the foreign mixture is called Biuret solution, which has a blue color. So this test is done to identify the presence of proteins and or polypeptides. So if you have a positive result, what does this mean? Move. Results. A positive results, we have a violet color appears, indicating the presence of proteins or peptides. Both of them, either protein or peptides. We can't confirm whether a protein or polypeptides unless we do another test. We will study it in the next video. So positive result, a violet color. So whenever we have a violet color with the Iodine test, this means that we have either protein and slash or polypeptide or peptide. They are the same. Okay? While the negative result, meaning that we don't have proteins and or polypeptides, and the color will stay blue. So there is no protein slash and slash or polypeptides. So repeat this. If we have a violet color with the violet, meaning that we have proteins and or polypeptides. We don't have, so we don't have neither proteins nor polypeptides. So blue, it's a negative result, no proteins. Deep purple or violet, positive result, we have proteins and or polypeptides. So this is the Iodine reagent and this is the test done.
Proteins can be detected by the Burette test. To 2 cm cube of egg white solution, add Burette solution dropwise. Shake after every drop is added. What color changes do you observe? If proteins are present, a violet color develops. If proteins are absent, the solution remains blue.
Move to this table. Here in this table we have the food test. This is as a summary. The aim for this test, why do we do it? The steps, which is the procedure, positive, negative, analysis, and indication. If we add these two together, it's the interpret. So Fehling test is done to identify the reducing sugar. What do we have to do? We have to add equal drops of Fehling A and Fehling B. And then we have to heat the contents of the tube gently over the Bunsen burner, which is the source of heat. Positive, we have brick red precipitate. Negative, the solution will remain blue in color. So after adding equal drops of Fehling A and Fehling B and heating the food, or you can say after applying Fehling test, brick red precipitate appears. What does this indicate? This indicates the presence of reducing sugar. So this is for Fehling test. Whenever we have brick red precipitate, this means that we have reducing sugar. Analysis, we can say after applying Fehling test, or after adding Fehling test solution, or after adding equal drops of Fehling A and Fehling B. Moving now to the test, iodine test is done to identify the presence of starch. What are the steps? Add few drops of iodine solution in a test tube that contains the food to be tested. Positive result, dark blue color will appear. Negative, no dark blue color. The color will be orange, yellow, or brown. Either orange or brown color. So analysis, after adding iodine to the food, dark blue color appears. This is analysis only of the positive. Okay? So this indicates the presence of starch in the food. While if we add iodine test and we have negative, so after adding iodine to the food, no dark blue color appear. This indicates the absence of the starch in the food. So here, analysis of the positive result. So after adding iodine to the food, dark blue color appears, indicating the presence of starch. Third test, this is the last test, the Burette test. Burette test is done to identify the presence of proteins and slash or polypeptide and slash or polypeptide. So add equal drops of Fehling A and Fehling B. No, we don't say Fehling A and Fehling B. We say NaOH and CuSO4. So NaOH is sodium hydroxide. CuSO4 it's copper sulfate. Positive result, violet color appears. Negative, the color stays blue due to the color of CuSO4. So after adding equal drops of NaOH and CuSO4, or you can say simply after applying Iodine test, violet color appears. This is the analysis indication. This indicates the presence of proteins and slash or either proteins and polypeptides, either protein or polypeptide in the food. This is the three tests that we identified them in this video. Okay guys, study well these notes in the table, and you have to know the positive and the negative results of each of the tests and their indications. Thank you. Bye bye.